our lab works with two major focuses. First one is to understand the mechanism of cell division, and second one is to understand the mechanism of development of antimicrobial resistance. Bacterial division is never been targeted for drug development. Understanding of bacterial division will help us to develop novel antibiotics. Briefly, after the completion of DNA segregation, bacteria divides using a machinery called as divisome machinery. It is a dynamic machinery composed of at least more than 2 dozens of proteins, and our lab focuses to understand the dynamicity of this complex. We have shown that the divisome complex can also regulate the septal cell wall synthesis and bacterial motility. FtsZ is a major component of this complex which initiates the division process, and several of our studies show that FtsZ can be targeted to stop bacterial division and thus the bacterial proliferation. In our lab, we also investigate other proteins in the divisome complexes. So other proteins such as FtsE, MinDE system, ZapE, FtsA, etc. So currently, antimicrobial resistance is a major concern worldwide. In our lab, we are focusing to understand the mechanism of the development of the multidrug resistance in bacteria. The major reason for the development of the multidrug resistance are efflux pump activation, biofilm formation, and the bacterial persistent formation. When efflux pumps are activated, bacteria efflux the antibiotics out from the cell. This increases the antibiotic requirement to kill the bacteria. Similarly, biofilms forms a protective layer around the bacteria, thus making it impermeable to the antibiotics. So work from our group is already published in several globally reputed journals such as Journal of Biological Chemistry, Lancet's eBiomedicine, Antimicrobial Agents and Chemotherapy, etc.
So my research work focused on the understanding of function of FtsZ, which is the core protein that organizes cell division in bacteria. So for this, I use techniques like protein-protein interaction, protein purification, biochemical assays, and microscopy. So the whole goal is to understand the binding sites of FtsZ from different bacterial species, so that we can target FtsZ to develop antibiotics that can target cell division in bacteria.
I am working on bacterial cell division. Here I am working on a protein which is important to bacterial cell division. Here we are proposing this protein as a antibiotic target. We have elucidated its role in different bacterial cellular processes, and which can be used as a target for antibiotics.
We have identified some of the molecules that can target both the efflux pump and biofilm formations. So the various microscopic analysis, biochemical and molecular mechanism, we are trying to develop some of the inhibitors that can target the pathogenic pathways of Staphylococcus aureus and the other escape pathogens.
Bacterial persistent phenotype is one of the most phenotypic variants by which are multi-drug resistance and which can generate different type of chronic infection. At the same time, I am trying to decipher the role of bacterial toxin-antitoxin system in generation of persistent phenotype and bacterial programmed cell death, so that we can get rid of this persistent mediated chronic infection.
Their lab works with multidrug-resistant bacteria and antimicrobial resistance. The thing that fascinates me more about their lab is their work on biofilms. So I'm trying to use my expertise of using free-living amoeba to eradicate biofilms. So right now, my expertise is on host pathogen interactions, and I'm using that expertise to combat the threat of biofilms produced by different and their bacteria.